Today we are going to start to talk about the phylum Chordata, and we are going to discuss many things under the phylum Chordata. We are going to give the characteristics of phylum Chordata. We are going to talk about the division under the phylum Chordata, and we are going to discuss one of the classes under the phylum Chordata. So let's start with the characteristics of Phylum Chordata. Under the Phylum Chordata, organisms here, they have four main characteristics that is common among all of them. One, they have a notochord, and this notochord, and some of them, it is present only around the tail area during the larval stage, and some of them, it is present throughout the length of their body, and it persists throughout their lifetime. That means it is present throughout their lifetime. And some of them, the notochord will be replaced by the vertebral column. And we have the dorsal hollow nerve cord that in some of them it has to form the brain, the spinal cord, and other parts that are part of the nervous system. And we have the pharyngeal slit that form the opening into the pharynx. And we have the post-anal tail. The tail is any part beyond the anus opening. So now let's talk about the division under the phylum Chordata. We have the subphylum Urochordata. Their main characteristics are: one, they have notochord that is limited only to the tail region. That means the notochord is present around the tail region, and it is present only during the larval stage. The organisms under here, they are surrounded by what we call tunic, and these organisms are called tunicates. The other subphylum that we have is Cephalochordata. For them, the notochord is running along the length of the body, and the notochord persists throughout their lifetime. And the dorsal hollow nerve cord that they have, it forms a primitive brain. The organisms here are called lancets. And we have the subphylum vertebrata. In here we have organisms like agnatans, fish, sharks, and tetrapods. Now let's take the subphylum vertebrata and divide it further. So under vertebrata, we can first divide the organisms based on jaw, whether they have a jaw or not. So if they lack a jaw, they are referred to as agnata. And under the agnata, we have one class, that is the cyclostomata. And we have those that have jaw. They are referred to as gnatostomata. They bear jaw. And under it, we have two superclass. We have the superclass Pisces that they bear fins, and we have the superclass Tetrapoda that they bear limbs. And under the superclass Pisces, we have two main classes. We have the class Chondrichthyes. In this case, the Pisces here, their skeleton is made up of cartilage. And we have the class Osteichthyes. In here, The Pisces here, their skeleton is made up of bones. And we have classes under the super class tetrapoda. We have four classes. We have the class amphibia. Organisms under amphibian, they experience a dual life. They can live in water and they can live on land. The word amphi means both. That means they can live both on land and in water. And we have the class reptilia. Reptilia, they are mostly terrestrial. That means they are living on land, and for them, they have a scale on their body. And we have the class aves. The aves, they have feathers which they use for flight. And we have mammals, class mammalian. They have what we call the mammary gland which secrete milk. And the organisms under this class mammalian, they give birth alive and breastfeed their youngs. The class amphibian, reptilia, and aves, they lay eggs. They don't give birth alive to their young one. So let's start with the superclass Pisces. So under the superclass Pisces, we are going to start with the basic characteristics of organisms under this superclass Pisces. So we are going to start to talk about the main features of the superclass Pisces, in bracket fishes. So in here, number one, they are aquatic animals. That means they live in water. Number two, they have streamlined body. That means at both ends, at the head part it is narrow, at the tail part it is narrow. As you can see here, narrow at both ends. And they swim with the help of fins. So what they use for movement is called fins. Next, they are cold-blooded animals. That means their body temperature, it changes continuously as the environmental temperature changes. For them, they don't have a constant body temperature as we have in humans. Next, their body is covered with scales. For them, what is covering their body, it is called scale. And if somebody asks you what is a scale, if it is for interview, you have to first specify in what subject. Because scale in physics is different from biology, and scale in economic scale of preference is different from what we have here in biology. So you have to specify it first under which criteria that the person is talking about scale. And the next one, they breathe through their gills. For them, they don't have lungs. What they use for respiration is called gill. And they give birth by laying eggs. Animals that give birth by laying eggs, as we have on the slide. Animals that give birth by laying eggs, they are called oviviparous animals. So these are examples of organisms under the superclass Pisces. We have the ray, we have sharks, we have dolphin, and we have many that are not even on this slide. So just like what we say under the division that the superclass is divided into two classes, we have the class Chondrichthyes and we have the class Osteichthyes. So let's talk about the differences between the two, how you can identify a chondritis, how you can identify, and Osteichthyes. So now let's talk about the differences. Chondrichthyes, they possess a cartilaginous skeleton. That means their skeleton is inside and it is made up of cartilage. And for Osteichthyes, they possess a bony endoskeleton. That means their skeleton is inside, as endo means inside, but the skeleton is bony. It is not made up of cartilage. Number two, the mouth is located ventrally under chondrichthyes. When you observe the mouth here, in this organism that we have here under chondrichthyes, the mouth is below here and the mouth is not located at the front part, at the terminal end of the fish. So in this case, but for Osteichthyes, the mouth is located terminally. That means the mouth is located at the terminal end of the fish. And number three, the gills are without operculum for chondrichthyes. When you observe here, in here they have what we call the gill slit around here. We have the gill slit around here, which is the opening into where we have the gill. But in here, the gill is covered by operculum for Osteichthyes. If they ask you to give observable differences between the two, what you see is what you have to give. Don't give things that you cannot see it. For example, the ear bladder, you are not seeing it. But if it is open, just for you to give differences between the two, you can give anyone. But if it is for observable features, what you see is what you have to give, like the operculum present here, not present here, the mouth that is ventral in the chondrichthyes, and the mouth here that is terminal in the Osteichthyes, that one you are seeing, then you can give them observable features, what you can see. So let's continue. Number five, fertilization in chondrichthyes, it is internal. That means it is occurring inside the body of the organism. But for Osteichthyes, fertilization is most often external. That means it is occurring outside the body of the organism. And fertilization is the fusion of the nucleus of the male gamete and the nucleus of the female gamete to form a zygote. And the skin here is covered with placoid scales. This one you can also give it for observable characters because it is outside, you can see it. And here the skin is covered with cycloid scales. Now let's talk about diagram of a tilapia, because this one is the most common one that we have, which is present across West Africa here. So most often they focus to ask you on tilapia. Let's start with the parts that are labeled in here and we can talk about parts that are not labeled. Let's start from the fin that is at the top, is called the dorsal fin. And we have the fin very close to the operculum. It is called the pectoral fin. And we have the fin that is found on the ventral part. And this fin is called the pelvic fin. And we have a line here, this dotted line running across the length of the fish. It is called the lateral line. And we have a fin that is closer to the anal pore. This fin is called the anal fin. And we have the fin that is right around the tail area. It is called the tail fin, or you can call it the caudal fin. And lastly, for those that are labeled, we have the operculum, the one that covers and protects the gill. Now let's talk about parts that are not being labeled. So we have the eye that is not being labeled, we have the mouth that is not being labeled, and we have the anal pore that is not being labeled, which is very close to the anal fin. So for your exam, you are expected to know the function of the parts, like the operculum that protects the gill. We have the eye that is there for vision. We have the nose, which I almost forgot, that is responsible for smelling, not for respiration. And we have the mouth that is responsible for feeding and respiration. And we have the fins that are responsible for swimming. And we have the lateral line that detects vibration in water. Sometimes you will be asked to state the sensory organs that we have in tilapia or fish. Now, one, you have the lateral line that detects vibration. You have the skin that detects touch. You have the eye that is responsible for vision. And you have the nose that is responsible for smelling. Now let's talk about adaptation of the fish. Now what are the things that is helping this tilapia to survive in its environment? Number one, it has an eye for vision. Number two, it has lateral line that detects vibration in water. And number three, it has operculum that protect the gill, which is used for respiration. Number four, it has fins that is used for movement. And the number five, which is the most common one that they use to ask students about, is their color. The dark color that they have on the dorsal part, that means at the top, and the light color that they have on the ventral part, that means on the belly part, the part that is below. Let's talk about how this dark color at the dorsal part and the light color at the ventral part help this tilapia to survive in its environment. This one is a very common past question that has been coming, so you have to take note of it. This dark color that is at the top, it blends with the dark color that is at the bottom of the ocean when viewed from above, which will make very difficult for the predator to identify it. And it has a light silver color at the ventral part. This makes the predator very difficult to identify it when viewed from below, because the color here, this light silver color that is below, it resembles just like the color that is above. So in that case, the predator may find it difficult to identify it. Next on our slide is the diagram of a tilapia with a cut gill. So after you have removed the gill, what you see is what we have on the slide. And you may be asked in your practical to remove the gill and draw it. And if it is a test of practical, you will be asked to draw the diagram of a fish with the operculum being removed from it. So this is what you have to draw. They will not ask you to draw the whole length. They will just ask you to draw the head with the operculum being removed. Now let's talk about respiration in tilapia. So now let's talk about how this process happens. First, when they want to take in the water, which they have to extract oxygen from it, they will close the operculum. When they close the operculum, they open the mouth. When they open the mouth, they lower the floor of the mouth and water will flow the mouth. After that, they will now close the mouth and raise the floor of the mouth, and the water will flow through the gill. As the water is moving through the gill, oxygen will be taken in by the blood vessels that are found within the gill, and carbon dioxide will be taken out into the water. So what happens here? Oxygen is being taken into the blood vessels surrounding the gill, and carbon dioxide is taken out of the blood vessels surrounding the gill into the water. After this process, the tilapia will open the gill and the water will come outside, and the whole process will start again. Now let's talk about the features of the gills that make them an efficient respiratory structure. One, it has a large surface area for diffusion of gases. As you can observe here, this is the gill filament. The gill filament, they are numerous, they are tiny, which provide a large surface area for diffusion of gases. And thin filaments, just like I said, for easy diffusion of dissolved oxygen. Now these filaments, they are not thick, they are very thin, which will make diffusion of gases very easy. If they were thick, oxygen will find it very difficult to pass through it, so they will be absorbed into the blood. Next, the gill is having moist surface area for dissolving gases. For gases to diffuse across a membrane or across a layer, they need to be dissolved in water, or they need to be moist for them to pass through. If it is not moist, they will not be able to pass through. And it is highly vascularized for transport of gases. So as the oxygen is being dissolved in them, the blood vessels will take them and transport them to where they are being needed, or to where they can be used. Lastly, you need to know how to draw and label the gill. So you have the gill filament that is red in color, and you have the gill raca having a tooth-like structure, and you have the gill arc on which the gill raca and the gill filament are attached to.